הסטודנט עם העבודה שלך נגיד במכון נשירה. אוקיי. אוקיי, לכם שאני יכול לעשות. מעבר. הייתי מעבר את התכנון קודם. לא הייתי.
Okay, hello to everyone. So, let's turn on the light. It's not working. Maybe from here. Okay. So, how are you doing? Did you start preparing already for the test? Some of you. So I want to say first of all that if you feel, because also I was a student, not recently, I am aware that some of you are postponing the study for the test for the last minute. So, how did they know? I had a feeling. I have cameras. They didn't tell you with their cameras all along. We see exactly what you're doing. Every time you type molecular neurobiology in your computer, here, I see it. I didn't talk about you specifically.
He looked at Tyler. He saw everybody was typing it. You didn't. You don't.
Yeah. No, but we can do also a rehearsal. If you want, we can organize sometime closer to the exam. If you want on Sunday or something like that, if you need or you feel the need to. So obviously there's a lot of material, and we're not going to go now over all the presentation of the whole year of the whole slide. So that's why I also told you that it's more effective if you come with certain questions about aspects of the material that you didn't understand, or you feel you need, or something in the vicinity. Yeah.
I'll start with a question.
Yeah.
So I was always confused how you really can classify something in the protein as tertiary or quartile structure.
Okay.
Because they're both super primary, secondary, that's clear.
Tertiary is still the same. There's the tertiary and like the fourth and the third level. Okay, it's always confusing, but the third level is still the same molecule. Okay, the fourth level is not the same molecule. So a domain is always inside a domain is a place inside that molecule, okay? But when we talk about primary structure, it just means something that is when we're talking about the amino acids themselves or the backbone, just the peptide backbone. That's where the primary stops. The secondary structure is U-turns, alpha helix, beta sheets. That's it. Everything above that relates to the same molecule is the third structure. And when we're talking about different molecules, if we're talking about the G protein, okay? So the alpha subunit and the beta subunit, they're two different molecules. but the interaction between them for the whole G protein is the fourth structure of the whole protein.
Global domain is still tertiary structure.
Yeah.
Okay, just everything is something complete, like different molecule, not domain, not subunit.
The fourth structure is different molecule. Yeah, it's interaction between subunits.
Subunits are always from different strengths. I mean, they're always. I mean, the definition of a subunit is
Mm-hmm.
different.
It's a different molecule. It's a subcompartment, or it's not a compartment. It's not. So you can have what we defined as a motif. Okay. So you can have several motifs inside the same molecule, and you could have also different like domains, but you don't have. But subunit is normally alpha subunit, beta subunit, which are like different genes actually. Okay, the different genes that are actually transcribing these subunits of the larger molecule. Okay, so that's why, yeah. Fifth, there's no like, I wrote the fifth structure? Yeah. Well, yeah. Well, the fifth structure is like, is everything. Okay? Like the ribosomal complex, we don't define it as like these are like the complex that you have in order to initiate translation. Then you have different proteins. When we're talking about different protein, it's a complex. So it's interactions between, let's say, a channel with all its subunits with a scaffold protein. So they form like this complex, or something. So it's like it's another level of organization. Okay, it's above that level of organization. It's for sure it's between different molecules, but not even that. It's between, because a protein, a lot of time you will almost never, like you'll find the subunit of a protein. Okay, so the fifth level is what we define like a functional level. Okay, so in the end, when we look at the protein as after we regard all its subunits together, okay, so we say, okay, this is a functional protein, it does a specific function. But that protein can play a role in one complex, and it can play a role in a different complex. And so, and obviously it undergoes association with other proteins. So these large complexes of associations, I somewhat maybe defined like as fifth structure. But in a sense, it's hard to define because no, fourth structure is very well defined. Okay, when you have you can have like specific types of receptors, and you will let's say like the nicotinic receptor. If we have the alpha 7 nicotinic receptor, so the alpha 7 is 7 subunits of the. It's composed of 7 subunits of the alpha, repeats of the alpha. But you can have the nicotinic receptor that is alpha beta, like it has 2 alpha and 5 beta subunits. Okay, so it's different compositions, and both of them are nicotinic receptors, and they function together. But the definition of the fourth structure is always like closed around that. There's not so much flexibility. If we say that any type of protein interaction is like fifth structure, then the protein. The fourth structure is what you define the protein. This is the protein. This is the enzyme.
Like different things that have been translated like separately and then always go together. That's what you define.
Yeah. They don't have to always go together, but the final molecule. The tertiary structure defines the final active functional molecule.
So that's the tertiary.
The fourth structure. I'm always confusing between the tertiary structure. A lot of times is not functional. Like just one alpha subunit of a channel, for example. Like if you have a channel, a lot of time we saw this channel that are composed of 5. This is how, if we look from above. This is like I don't know, we saw an example of a lot of channels look like this. so they compose of 5 different proteins, and this can be like 2 alpha subunits, and these could be 3 beta subunits.
But it's not different proteins, it's the same protein.
No, it's different proteins. Like you can have
All of them together are the one protein that is the receptor, or is it?
no, they're not. Okay, sorry. They're different. It's the same protein. Okay, it's the same. Together, they're the same protein. That's composed like, but they're made of different molecules. Yeah. Okay. This is force. Here, in the interactions that happen in the level of the same molecule are maximum their 3rd structure, and the interactions that happen here are the 4th structure. Forget about the 5th structure, okay?
You can have domains that are of 2 different molecules.
Well, a lot of times it depends, but for our case, yeah, you can have like, but it defines how you say domain, okay? But you're right that in this field it's a little bit problematic. A lot of time people use domains to describe features of a protein that can be because of association of two subunits, okay? But in general, when we say subunit, it's not different. So domains is problematic. Okay, like also like motifs, domains. A lot of the time people use them interchangeably. But again, I will try that the questions in the exam, in that sense. For example, I will not ask you about the 5th structure. Okay, and I will not. And if it will be like when I say like a subunit or something like that, that we defined in a good way, that's like that the protein is composed of different subunits, then you know that it's different molecules. Okay, so it's subunits of the same active protein.
So you said like that people usually use both motifs and domains in the same
Motifs normally. So in general, domains are usually considered like the same molecule. Okay, like part of the same molecule. But sometimes people can use it and say that it's not the same molecule. But for our sake, a domain is a part of the same molecule.
And the motif is not?
A motif is also a motif is like several motifs can compose a domain. Okay, motif is in smaller scale. When we talked about motif, we talked about like a calcium binding motif or a DNA binding motif. Okay, so it's like motif. Most of the time, a motif will be defined as something that has a certain function, and a domain can be something that just looks like something. Okay, it will be like a globular domain or a fibrous domain, and that is like a more linear or things like that. Okay, so in respect to that, we define for sure motif is inside the same molecule, and also domain is inside the same molecule. When we talk about subunits, different subunits are different molecules. Okay, and when we say a protein, we mean all the different subunits that are included inside this protein. Okay, so we can have
My actual question is not about proteins, but it's someone like more.
Yeah.
I just if you can go over the protein homology.
Okay. So you specifically talk about the question in the exam that I gave you?
No, something that confused me.
Okay. So yeah, I can put answers. Yeah, but it's best if you go over it by yourself and a little bit before that. You know, so let's say I'll publish the answers on Thursday. Okay. So I highly recommend to go over that before that. So actually, I will answer your question through this question. So here, between homologous protein, there is an identical sequence of amino acid, a similar sequence of amino acid, identity of secondary and tertiary structure.
B or D?
Is it an A, B and B? So, similarity?
So the thing about homology in general is similarity. Okay, we don't necessarily say identity, but if you see correctly, okay, so.
So yeah, I just meant like which one is either B or D or both.
So if we said that there are similar sequence of amino acids compared to, and also similarity in the secondary structure. Okay, a lot of time we say similarity in the sequence and similarity in the secondary and tertiary structure, so it's B and D in that case.
Proteins that they are homologous. Like what can you say about two proteins? I mean,
It can be.
proteins have similar amino acid sequences. The difference can still account for different secondary and tertiary structures, and also like accidental.
A lot of time you don't know the tertiary structure, and you say that that the sequence is homologous. Okay, but a lot of time, so the homology can be a lot of time. It depends if you say homology in the sequence or homology in the protein. But in general, both these definitions, and also this is what we discussed in class, that a similar sequence of amino acid and a similarity in the secondary and tertiary structure.
What can you say about, if I know the two proteins that are homologous, what does it mean about them? What sort of connection?
It really depends on the specific protein, but a lot of time, because of the homology, we can assume that they came from like a ancestral source. I mean,
Yeah, the actual DNA. I mean, if we assume that they both came from the same ancestor and one of them is like a mutation or something else, both of them are mutations, then the secondary, like the sequence should be very similar because the sequence is pretty much saying what's DNA.
It can be that the function stays the same, and there is a lot of changes in the sequence because it doesn't matter their function.
But then
just-
how do we know they came from the same place? They do the same.
homologous doesn't necessarily mean, that's what I told you also in class, homologous doesn't necessarily mean that they came from the same ancestor, okay? This is not what when we say homology, we actually mean similarity. And this is important that you know these two definitions, because there's also, people use homology also when they're not talking about evolution, okay? So, when you ask me like what does it tell us that they're homologous, I said that it indicates that maybe there is a common ancestry, but there can also be a common function that is not well preserved in the sequence, but these proteins will be considered homologous proteins, and that's why D is here. What you notice that you won't have.
Only D is also homologous.
Both are correct. B and D are both correct. So
But
that.
if two proteins only have a similarity in secondary and tertiary structures, and not in the sequence of amino acids, they're not homologous?
No, they are considered homologous.
Correct, which is correct.
Yeah.
Yeah, but what they
Yeah.
understand is that it's separately also.
Yeah. They're separately. It can be, yeah. So it can be both of them separately, and also you know that, like, we didn't put here. I didn't do this test first of all, but for example, you don't have here A and B. Okay, you don't have the option of A and B because this can be misleading, because you say also identical, identical is not homologous, it's the same. Okay, but you can say like maybe part is identical and that's why it's homologous. But for a reason they didn't put here A and B. because it's like misleading. But it's important to know that two levels of homology, one of them is the sequence and one of them is the actual shape of the protein. Yeah.
From what I understood in the rule, they say that the same primary sequence does not determine the same secondary and tertiary structure. But you can have the same primary sequence and still have different tertiary structures.
Exactly the same primary sequence?
Like you cannot determine the secondary structure from the primary.
You cannot determine, but it will have the same. Like it's hard to. We don't know. It's hard to anticipate what exactly are the foldings that will occur. And if you remember, we said that it's like a valley of energy. Okay, like it's like the lowest steady state of all the foldings of the backbone will lead to one conformation, to one final conformation. But obviously, if you have chaperones there or don't have chaperones there, it can cause like misfolding. But for our case, if we're not talking about different conditions or pathology or something like that, if we have the same sequence, we'll have the same folding or the same final structure. Yeah. Well, always, it's never always, but for our case, it's always. Yeah.
A general question about the test.
Mm-hmm.
Usually, like we know, like a few answers are like one answer is like probably right,
Mm-hmm.
two answers are probably wrong, but then we have like really like two candidates or choose them. Like not sure what to choose. So if we have, if the right answer is kind of a combined answer, in this case B and C and D, and we choose an answer which is partially right, do we
Mm-hmm.
get credit or not?
It's a good question. It's a good question. I need to discuss it with my supervisor and exactly how they did it in the previous years. Not to do, but don't take my word for it. But I guess that you will get part of the. I guess that you will get part of the grade.
Can we just comment, like, it might be most questions not, but if something's like confusing and we're not like exactly sure what the sentence means, so we can say like A if you mean yada yada yada, and B if you mean something else.
No. In general, no, because it gives you like an option for me to choose what is correct. So you don't pay for something. In that sense, you don't pay for something that you said that is not correct. And so, but first of all, I will be here, so you can ask me. And second of all, if we see again, like I said, if we see that there's a drastic, massive failure in one question, then normally we will not consider that question. Okay, so if there's a question that is really problematic in terms of how we define it, then a lot of times. But I'm really working hard on making the question as straightforward as possible, not misleading you and stuff like that, and really related to what we learned in class. Yeah.
I have a question about the bonds. In the presentation, you mentioned that ionic bonds are not, there are no pure ionic bonds, there have to be covalent. I don't understand.
Meaning that there is always a level that the electrons are shared. Okay, it's not, there's no like bond that you have only when you have interaction like NaCl, like or something like that between these two ions. Then you will always have a level that the electron will not be purely, the electrons will not be purely at one of the atoms. It will be always a level that some of the time the electrons will orbit also around the second atom. So it's not like so that's a level of covalent bonding. Covalent bonding is defined, right, because we have the shared and they're revolving around, they're orbiting around two atoms. Yeah, it's two. And it's an electron pair, okay? So the time you have that, but in ionic bond, because of the electronegativity, like very extreme electronegativity, then you would have the electron will be most of the time they will be around the electronegative atom, okay, and there will be less time around the non-electronegative atom. So it will be like a weaker bond or weaker sharing, but there will be still a level of covalent bonding there. So
Why does it have to be 2 electrons?
I don't know. Like this is like
Hydrogen, like, only giving 1 electron.
yeah, but when two, like let's say we didn't talk about it, but let's say you have 2 hydrogens that are bonding, then one is giving 1 electron and the other one is giving the other, and the 2 are orbiting both of them. Okay. Yeah.
Could we just like question 3 versus 4 after like the thing which I was struggling for? Okay, like a double bond, a carbon chain, fatty acid will cause. Okay, it will cause bending. Yes, because yes, on one side we have this double chain and you can't have this zigzag chain bond.
Okay.
But I'm not sure about the flexibility or about the rigidity.
When we talked about the double bond in a carbon, one of the things that we said, it's apart from the fact but the stability, it's actually like we drew it also. So if we have a carbon here and we have a carbon here, and let's say here we have hydrogens, for example. So a double bond in general is like you would imagine it, like you would have 2 rods, metal rods, that are here between them, and it doesn't allow flexibility in that space. So the fact that you have like more double bond, so it will cause a rigidity.
Any double bond, right? Any double bond doesn't allow you.
Yeah, any double bond. So it for sure is not B. Okay, so it can actually cause. So it can cause also bending, and it can cause also straightening in its region, because you can have like we saw. Sometimes if you have a cis, if you have like you can have like when we maybe I'll go back to what we saw in the. Well, 3 answers are right, so what can you... So
Rigidity and bending.
So it can cause... First of all, so again, this question is a little bit problematic and I will not give it, just because of what you said, but in general, if you have a double bond, it depends how it's closed, okay? So if it's like in cis conformation or in trans conformation, then it can cause. So it depends if it had a kink before, like if you have a conformation that is going like this, and now you would have, and here you have like a. So it's not. I'm just showing the presentation because I can't draw it. No, the bonds are not defined like if you don't have a double bond, then you can't define it as a. Then it's not defined as cis and trans. No, you don't have the fact that you don't have trans bond naturally. It doesn't mean that in this case. So that's what I told you about fatty acids in general. That you don't have trans conformation normally produced in nature. You normally have only cis conformation, meaning that the molecule will look like this and will not look like this. Okay. What? No, I'll show you in the presentation. It's better because I'm not. No, it's not here. Protein structure. No, it's not protein structure. Biochemistry. No. Okay, maybe I have. Okay, so in this case, so in this case, the double bond will cause bending. Okay, so we have like here, everything is without double bond, and you have a double bond here, which is, and it will cause a bending. Okay, of this area. So again, it defines what you define as a region, because in this case, if you have a double bond and this double bond is like linearized, or this cis double bond is converted to a trans double bond, okay? So it's bent, but in general it depends. That's what I said about this question, because it says around its region, okay? So it depends how you define the region, okay? If you define the region as a large region, then it's hard to define. Then if the region is large, it can cause a straightening. Some people would say that I don't know, that if you had like another double bond here that did like, and the molecule would go like this, and then you would and the fact that that you have like, if you have like two trans bonds, then maybe it will cause like a straightening of the molecule. I'm just saying that in general the correct answer is it will cause what? No, you're right that it's right that the correct answer here is it will cause a bending in this region and a rigidity in its region. So it's A and C in this example. But when I'm saying that, I can understand why you can get confused by the fact that we learned like that all that trans are like straight. The trans molecules are straight, but they have double bonds still inside them. Okay? Yeah. No, you're right that. So let's say we have a molecule. That looks, okay, that looks something like this. Okay, and now we present a double bond here. You have a carbon here, and you have a carbon here, and you have a carbon here, and you have a carbon here. Okay, so now you introduce a double bond here, and the double bond is in trans conformation. So you'll have now something like this. Did it cause a straightening or it caused a bending? So in general, if I would ask a question like that, I would say between the two adjacent carbons, for example, and then it will be like obvious, okay? Or between the strands of or I would define like a molecule that I would say we have this molecule that looks something like this, and now we introduce a double bond. What will it cause? Okay? In that case, it will be more clear. Okay? I'm just saying it's right that the answer here is A and C, but I can imagine again when I'm coming from your perspective, and I'm trying to do that, to see where you can get mixed up here. Yeah.
So, according to the description of this test, A and C can't be right.
Why?
Because it says just one of the options A, B, C, D, E, F, G, H can be right.
Yeah. F?
Yeah, if F is right, then G is also right.
Fine, but it just made a mistake.
So
Well, you see, that's a bonus question. You see this thing, you mark A and C, and that's it.
So that contradicts the assumption that
Like I told you, I didn't write this test, so they made a mistake. Yeah. Yeah.
Why connection between the ion and the water molecule
Not hydrogen, you mean? Between them, again, between ions and water?
Yeah, because in the presentation,
Mm-hmm. It's not considered, so did we say ionic interaction or ionic bond? Ionic bond? Okay, actually, in the first one, right? Interaction. It's not bond. Yeah, I know that here.
I think in your questions,
And the answer was ionic bond.
What between? So maybe it was a mistake in the. I think that this year. I think that in previous years it was ionic bond here, and that's also why it has ionic bonding here.
the question
But I think it's I remember specifically that I changed it to interaction because of that. Okay, because there's no sharing of electrons between the magnesium here and the oxygens here. Okay. No, you have a ionic bond, for example, in a salt, like NaCl.
But here
So
it says ionic interaction results from the attraction of a positive charged ion or a negative charged ion. So it's kind of a contradiction because the water is not negatively charged or positive.
yeah, but it's local negativity. It's local. The charge here is a local effect. Okay, it's the orientation. Actually, these two are not referring to this. Okay, so these are talking about ionic interactions, and here in aqueous solutions, iron anhydrate is surrounded by a stable shell of water molecules.
Yeah, but in the question,
what's the bond considered? What's the interaction considered? Hydrogen or
The bond here is ionic.
water and
Ionic. It's ionic interaction. There's no hydrogen here. Okay, the hydrogen are here. They're not involved in this interaction.
isn't it hydrophilic? That's the meaning of hydrophilic attachment,
No, hydrophilic is a hydrophilic interaction a lot of times, or the hydrophobic effect, or it depends. I don't know if we said like hydrophilic. We defined hydrophilic interactions. We defined hydrophobic effect, which is important, which is this, which results from the interactions between the water molecules themselves. Okay? What? The fact of a ionic interaction?
Yeah, because you defined it one way and then you asked what type of chemical interaction exists between Na and water, and the answer was ionic.
Yeah.
Kind of
What else? Why?
because you defined ionic interaction as the interaction between two ions, and water is not.
No, if I define ionic bond,
In the presentation it says ionic interaction.
of a okay, so so I understand what you mean. It was probably better like this is a second type of ionic interaction. You're right, like that. In that sense, it's confusing.
Wait, so if you have a negative ion,
No, if you have interaction, what we defined as hydrogen bond is the interaction between hydrogen, okay, and an electronegative atom, which is, in this case, it's oxygen, but it can be other atoms as well. This is not ionic interaction. This interaction is hydrogen interaction.
if you have a negative ion and
Mm-hmm.
so you don't consider it as
You mean because of the what? Because you mean because of the it's it's the size. You need to define the actual bond that happens there. Actually, this relates to what we talked about when you asked me before about what defines like ionic interaction and what defines like covalent bonding and what is the degree between hydrogen bond and covalent bond in general. Okay, so also in hydrogen bond, it's a type of, it's not a covalent bond because there is no like sharing. There is like a sharing in a sense, but it's like a degree of how much the electron is shared. Like in ionic, it's like the electrons are almost, when we're talking about ionic bond, and we talk, so it's like the weakest type of sharing of the electrons together, of the two atoms together, and then you have the hydrogen, and then you have the covalent bond. And when I said that ionic interactions in general, then these are ionic interactions between two molecules are mostly to describe that you have some kind of attraction between something that is positive and something that is negative. It's not a sharing of electrons. Okay, there is no sharing of electrons in that process. What do you mean? Like? Mm-hmm. Based on the fact that you're supposed to know that that the water molecule is polar, and the way that it attaches a positive ion is this way.
Yeah, but if it was a negative ion, then you attach to the hydrogen, and then what do you do? And you have hydrogen on the one hand and an ion on the other, so how do you decide which, what interaction is that?
No, you're right. You're right that it's confusing, and it's actually a spectrum. Okay, so it really depends. In some case, you can have. So, like you said before, if you'd have an oxygen there, the thing is that when we talk about that's what defines like electronegativity and actually charging. Okay, and molecules that are charged. In the case that we have hydrogen bonding, then you don't have really an ion on the other side. Okay, you will not see an ion on the other side. You have another molecule, another atom that is electronegative, but it can be totally like another water molecule. So it's totally balanced. It's not, the net charge of the molecule is zero. It's neutral. So the hydrogen bond is in. This is how we define like a hydrogen bond in general. You'll not see like a hydrogen bond that is attached to an ion. Okay? Then chloride, it covers. So there's no hydrogen bond between the chloride. It's just that the water molecules will cover the molecule, like surround and create like a. They will be hydrated around, but just in the reverse orientation. Okay, so but we say ionic interaction, but
So can you define again hydrogen interaction?
We didn't say hydrogen interaction. Okay.
Hydrogen bond is what kind of interaction is a hydrogen bond?
So, like it says here, a hydrogen bond is the interaction of a partially positively charged hydrogen atom in a molecular dipole with an unpaired electrode from another atom.
Ionic interaction?
No.
The other guy is
The
not.
other molecule is not charged.
If I have an interaction, like I have an electronegative molecule and an electropositive molecule, and they attract each other, the net charge of each one of them is zero. None of them is like an ionic molecule.
Yeah, you may not have. It doesn't have to be a hydrogen there.
So that can be counted as an ionic interaction? What kind of interaction is that?
I think no, this is not an ionic interaction. This will be like like a dipole interaction or a charge separation interaction, like even a magnetic interaction.
You have to include one element, either a molecule or an atom, that the net charge of it is not, is like negative or positive.
Yeah, normally when we talk about, and not to complicate things too much, when we will talk about ions, we'll talk about like atoms that are missing or they have more than one of the electrons, meaning that an atom that has a net charge of negative or positive. I will not call a large molecule an ion. Okay? So if you have like a small molecule, like the ions that we all know and love, like potassium, sodium, chloride, calcium, all these are ions, and their interaction with water will be an ionic interaction. It will not be a hydrogen interaction. Okay? So hydrogen interaction, and also specifically, I will ask you about hydrogen interactions in the context that we learned. Okay? I will not ask between, like, we talked about hydrogen interactions in DNA, we talked about interactions in alpha helixes, we talked about hydrogen interactions beta sheets, okay? And we talked about hydrogen interactions between water in themselves.
So hydrogen
Bonds.
are a subtype of
Covalent.
generally of covalent bonds.
It's in a sense, okay, so it's a degree. It's a degree of how much the pair of electrons are shared, how much time they're spending around both of them.
So
If it's a carbon,
bond, how much time does it spend?
so most of the time it spends around the oxygen, but it's not like an ionic bond. In an ionic bond, it's almost, yeah.
Like the covalent bond is between the oxygen and the hydrogen in the water molecule.
But
And then
there is a covalent bond there?
Outside the it's between two molecules, so it's different levels.
Right, because it's a different degree. This results, and we didn't discuss that, but this results from the amount of free electrons that are available for sharing, and also it's not the degree of the possibility of these electron pairs to be shared is different. So in oxygen, it has like two connections that are stable, and these will form the covalent bonds, and it has two electrons that are like more free to do like other interactions. But these interactions will be weak. For example, you will not see an oxygen. It's not like a carbon. In the sense of a carbon, it can form four covalent bonds. Oxygen will form two covalent bonds. Okay.
And then if an oxygen, like we have a water molecule and a different hydrogen atom, then the oxygen will form a hydrogen bond with the hydrogen.
Yeah.
So
it's attached to two. It's attached to two. In a water molecule, you have two covalent bonds, and you have you have still two electrons that this oxygen, in a sense, is not willing to share anymore. Okay? The oxygen had two electrons, and he shared them with the hydrogen. So it started like you can look at it in a sense. If you have this oxygen, so this is completely wrong. So I hope they're not they're not filming this. But let's say this is an oxygen, okay? And you have circles and orbits of the electrons that are circling around it, okay? So let's say it has two electrons here, and it has two electrons that are spinning around here. Yeah, I'm saying, okay. For the sake of the intuition, I'm not going to teach you about valence shells and stuff like that right now. But, so if you have another hydrogen here, and it has its own electron, then it will give, so let's say they will start sharing two electrons from here, and you have another hydrogen that has another electron, and it will take this electron here, and they will start sharing this electron. Okay? And then you will have two more, and I'm not talking about exactly how, because it's not even, these orbitals are not also correct. Okay, so they can have all sorts of, they don't spin like in a circle, they do make all these shapes, but never mind. In a sense, these two electrons, this oxygen is not willing to share anymore, but they can still interact. And the interaction that they form, but in the interaction that they form with the hydrogen of another molecule will form the hydrogen bond.
And then they will still be around the oxygen?
They will also spend some time around the hydrogen that is shared. Okay. Yeah. The covalent, this bond is very, very strong, the covalent bond, and the hydrogen bond is much, much more weak.
So it's not a matter of the electrons that they share that will be closer to the oxygen. I mean, that will spend more time around the oxygen, and that would make the dipole. I don't understand. No, I don't understand at all.
In all of these, okay, also in this bond, these electrons will spend more time around here, and this will form this dipole. Okay?
That's what's forming the hydrogen bond. I mean, that's what's causing the... That's what I understood.
It forms a dipole, but you still have interactions with the... So apart from these, so like you said, in this example, we see it in a better way. So like you said, there are six electrons that are free to interact in the oxygen. Two of them are doing this covalent bonding, okay, but still you have still they would spend more time around this region, and that's why you get this dipole. But these electrons that are free to form hydrogen bonds, so the dipole, in a sense, also the dipole and the hydrogen bond result from the same phenomena of the electronegativity, okay? But in a sense, and also, again, not to complicate things too much and getting into these, to the physics, but you have two bonds that are stable, two electrons that are dedicated to the hydrogens, and they're covalently linked between them, and you have two electron pairs that are available, but they're not available in the sense that they can form new covalent bonds like in the carbon. They're less available, but they can still form interactions. They can still spend times, like in this case here, between this oxygen and this hydrogen. Okay?
They're still in between, like, my part where I'm still, like, when I still understand this is they're not really shared with the oxygen. Like the oxygen doesn't really share its electrons now with the hydrogen. Only they spend, like, in the orbital shell around the oxygen, they spend more time closer to the hydrogen because they're attracted to the positive charge there. Like,
look, they are shared in a sense, but I don't want to get into that too much, because this is matters of, like, how much time does it spend around. I think I put you, there was a picture of this interaction. In the end, I think I didn't put it. I'm sorry I didn't put it, but...
Would you say that a hydrogen bond is a subtype of electronegative bond, which is not necessarily true for covalent bonds? I mean, covalent interactions can also happen when the two sides are, their charge is 0.
Yeah, but again, okay. I don't want to, on purpose I don't want to answer that too much, and I don't want to get these definitions too much, because it's beyond the scope of what I'm trying to do, okay? So the idea is, it's hard to get into this definition of these bonds, because there are complex interactions that are happening also because the identity of the atoms, like different atoms can form, and different electronegativity. But again, here in this course, this is not my objective. My objective is that you will learn and also not that you will learn, like, the hydrogen bonds that are specific also to biology or to molecular biology. And if you get a general intuition of that, that's very good. but it's more important to learn the specific type of bonds that we talked about in class. Okay? Like the specific type, it's more important for me, so I will never ask you about what we talked now, about the sharing of the electrons. I will never ask you about like how many electrons are available for interaction between the oxygen, and how many of them are dedicated for the covalent bond, and how many of them are dedicated to hydrogen bonds. Although I can ask that from this, but this is not my objective. Okay, my objective is for you to be acquainted and to understand the type of bonds that are happening in biological molecules.
Only fixed strength-wise. I mean, that is
Yeah.
related to the biology. Like covalent is the strongest.
Yeah.
But ionic and hydrogen, which is like more stable.
So you have this slide over here, which is important. Did I pass it? Where's the thing? Okay, so when I mean electrostatic, it's usually ionic. Okay, so hydrogen bonds, and then you have covalent bonds, and these are non-covalent interactions. Okay, so there is no equal sharing from this point on. There is no equal sharing of the electrons between the two atoms. Okay. So this is like or no strong sharing, but like you see it here, it's a scale. Okay, so there's always it's not like something that is clear cut. Like you're right that between a double bond and a single bond, then you have just more electrons that are shared. So this is more dichotomic. But the sense of which how much an electron is shared, that's why I also said that in ionic bond, there's always a degree of covalent bonding. There's always a degree of sharing of the electron. It's not purely ionic. Okay? Yeah.
So, is there a limit to the amount of hydrogen bonds you can form?
Every atom has a different capability, and that arises from so in oxygen it can almost only form 2 hydrogen bonds because it has 4 electrons that are available to form hydrogen interactions. Okay, like we saw here, and we saw also examples of the hydrogen interaction between the amine groups in the amino acids, and over there I think it's only 1. So here it's every oxygen can have, like here, because there's a double bond between this ester group, then it has only one option. The oxygen has only 2 electrons that are like free to form this hydrogen interaction, and in water molecules it has 2. Okay? But again, I will not ask you about, I will not get into detail. If you have an ester group and a double bond and oxygen, how many electrons are free to form hydrogen interactions with water molecule. I will not ask this question, because this is not in my sense, this is not the point. Okay, the point is to know that there is this interaction, to know the scale is very important. Okay, where is it located? That covalent bonds are the strongest and double bonds are stronger than single bonds, and that electrostatic interactions and van der Waals are weaker interactions than hydrogen bonds. Okay, that is important because it's important to understand interaction between proteins. Okay, for example, like here. Okay, so again, here you see like ionic bond or ionic interaction. This is like a problematic way to show it in general, but also I think that here they mean, I don't know if they mean positive of this molecule or an ion of this molecule. But let's just say that I don't like the definition of ionic bond and interaction in general. Okay, partly because in order to understand it, you need to understand a little bit about the electron sharing and stuff like that. Okay, so it's important to understand the that there are these different connections and their strengths. So ionic interactions that are electrostatic are weaker than hydrogen, and hydrogen are weaker than covalent bonding, which happen inside the molecule and not between the proteins. And van der Waals interactions are like the kind of weakest interactions, the weakest interactions that we learned about in general. They're like these interactions that arises from, like I told you like before, like random fluctuations in the charge of the molecule. Okay, but still, like in the example of the gecko, a lot of these connections together can form a significant strength. Okay, but for example, it's important to know that van der Waals interactions are non-specific. There's not something special. Any two atoms can form van der Waals interactions. Any type of interaction that we're like, so there are no rules in that sense.
So what makes the gecko a lot of them, like if it's random, it can happen in any kind of...
That's why it can climb on any surface.
So what, but why does it have a lot of them if it's something that is random?
How does the gecko know it and we can't?
Because van der Waals interaction largely relies on proximity. Okay? So, and when you look at something in the molecular structure of it, then the fact that it has a lot of surface contact in the gecko, so it has these carbon nanotubes in the tip of its toes, and these actually enable for very atomic proximity between the surface and the leg of the gecko. Okay, so even if you have, when you think about surface like contact, you would say, if I take a page and I put it here on the wall, then I have a lot of surface contact. But actually, if you take a page that is full with fibers and hairs, then the surface contact will be a lot stronger. You have a lot more surface contact, and also because it's proximity, then the leg of the gecko. So if you have, so any wall is not flat. Okay, molecularly nothing is flat. When we look at the nanoscale, everything looks like these ridges. Okay, what? Graphite, you will never see one sheet of graphite. You'll see like a lot of sheets of graphite. So in graphite it will be like these steps, right? So any surface in general is filled of these ridges. So the legs of the gecko, the hairs of the gecko can form these interactions, and this is what allows it to actually increase the contact with the surface to like a maximum, not to maximize straightly, but in a very efficient way, in order for these van der Waals interactions to happen. Okay, so van der Waals really depends on proximity. Okay, any type of bond depends on proximity, but van der Waals really decays the efficiency of the bond or the attraction really decays in the function of distance. Yeah, more questions. Yeah. Yeah. You always talk about questions that I don't like. Yeah, so hydrogen bonds can have varying distances, because also inside the hydrogen bond, it's because this is like an interaction or a bond, then you can also in covalent bonds between they can have like varying length or distances between the atoms. And the thing about linear is that, and this is like a blowout name to say that it's linear, but what we meant here actually is that if you have, if this is an oxygen and this is hydrogen, and then you have the bond, and then you have oxygen here and hydrogen, okay? So this is linear, and this is not linear. And what we mean, in a sense, in that sense is that the distance, the length of the bond is larger, so it's weaker. But again, I don't like this because it sounds linear. Just ask about the length of the bond.
Yeah, it's
Okay.
also like, I mean, it's used linear, we could just turn the
Yeah.
molecule.
Yeah, it just means, in this sense, it means the shortest possible hydrogen bond. Okay? Yeah.
Francesco, away from this molecule bond, but we call what the G-protein receptor
Yeah.
all these crazy
Things.
cascade bond activation steps.
Yeah.
We need to learn all the sub-steps with the names of the proteins who activates whom.
When we learned about specific systems in the G protein, and for example, you don't have to be like, it would be a very good bet to know for example, the pathway that activates cyclic AMP that we learned about also in articles and also gave an example in the class and know the steps that are going there. Okay, so, and also we learned, again, we didn't learn so many examples of G-protein copper receptor. We learned specific pathways, okay? For example, in the visual system, and we learned about the cyclic AMP, and in the case, for example, of the TABI gene, I didn't even tell you what's happening beforehand, just as part of it. But again, I would focus, I know that the part in regard, I'm sorry that I'm going back to the bonds because it's important for me to say that. Don't focus on things that we didn't talk about. Okay, so the only place where we talked about like what we discussed here for like half an hour is here. Okay, so when we said like this can form this and this can form this, like a lot of the types of bond that it can form. So this is important to know in general, especially carbon and oxygen and hydrogen. Okay, the amount of bonds that they can form, because we saw a lot of examples, and it's important for general knowledge. Okay, so know this, but you don't have to know anything about valence shells or available electrons or stuff like that, because we didn't discuss it and on purpose. Okay, so I would focus on and also you saw in the exam, in the example exam, you didn't have questions like that. Okay, so you should focus on general aspects that we talked about. But you should know, for example, what are the bonds inside an ATP or whatever, or like I told you, you need to know which bonds exist in famous molecules. Okay, like that we talked about. For example, if I ask you, is the backbone, what is the type of bond that exists in the backbone of a protein, or a backbone of an mRNA, and I say ionic bond, hydrogen bond, covalent bond, so you're supposed to know that. Okay, that's what you're supposed to know, and even you're supposed to, what? In the backbone, yeah, that's what defines the molecule. Yeah. So it's a trick question. Yeah, it's a trick, but an easy question. Okay. So yeah. But when we're talking about structures, about secondary structure and tertiary, quaternary, or we'll call it just. I think I'll skip the secondary and I'll just say first, second, third, fourth structure. Okay. Let's settle for that, so there will be no confusions. If there will be a question about that, I don't know. Yeah. I didn't hear. Do you know? Should you know something that's not related to the brain? What did we talk about? What did we say about the kidney? Ah. So again, the pumps. I think we always like gave an example also about things that are happening. We gave a little bit examples of pumps that are also important in muscle and stuff like that. But in general, we will also ask you like general question about pumps. But there are specific. I'm not saying that I will ask you. I will not ask you about specific pumps because there are specific pumps that are important for neurons, and we learn about them. No, I will try not to give you something that is a niche, okay? But in a sense, like you can look here about the question about how's it called, this chapter? Transport of ions. So you see, like here they ask like about coupling, like mechanism of action. How does this happen? Okay, where's the, in the sense, where does the energy for this transition come from? Okay, so for ATP, antiporters, synporters, ion channels, or any one of them, you're supposed to know, like, I think from this test, you're supposed to know everything besides this, question 26, which is not exactly what we learned. Yeah. What else?
Okay, so also the actual exam will be a bit easier than the test exam.
The actual what?
The actual exam will be a bit easier than the test exam. Example.
Why?
You always train hard and then.
No, but this is an example test. No, but what is missing here and will be in our test is a few questions about the papers that we talked about, like I told you. And again, with the papers, I will not ask you what is the name of the author, okay? And I will not ask you in the, so, although one of them is Kandel, but let's just say there are specific take-home messages and important things about the articles that I can ask about. Maybe. We'll see. But again, try to what we want to, again, you need to understand that what we want from you in this class in general is to be is like an introduction to the world of biology in general. So you need to know, that's the basic thing, to know the players, to know who we're talking about here. So if we're talking about synporters, antiporters, you have to know them. You have to know what is the mechanism and what they're doing. You have to know differences between DNA and RNA. You need to know proteins. You need to know amino acids, a little bit of sugar that we talked about. And we talked a lot about what RNA does, and we talked about, there's a large section about G proteins and a little bit about methods. Also here, in this test, there are not a lot of questions about methods. But there are methods that repeated themselves, for example, in a lot of articles, and methods that are very, very common, and we talked about them a few times, that I may ask about. For the method, yeah, I'll add it.
Can you repeat the explanation for electrophoresis or however?
Electrophoresis?
Yeah.
I think I said that. Did I say that it's included or not included?
No, not like in the last.
Yeah. But in the last one I said.
No, not for DNA only. Like it's been repeated a lot of times for proteins and.
Yeah. Okay. So for electrophoresis in general, is a concept of making biological molecules or pushing biological molecules through a gel, which is actually a mesh, okay, or a dense material. And what it actually causes, it will always cause, in a sense, the objective of it is to form a separation, okay? And the separation can be based on a few things. But the most common example that we saw in the sense of proteins was like Western blot, okay, or immunoblot. Sometimes it's called immunoblot, but also Western blot is that you take a protein that looks like that, and then you denature it and add SDS, which is a molecule that bounds to the chain of the protein. It causes it to be positively, negative. I don't remember, but it causes it to be charged in an homogeneous way, and and when you load the sample onto the gel, then
The most charged will go into the further.
in this sense we're not, when we're charging something uniformly, then the movement of that in a uniform electric field will be constant. Okay? So here the point is not the charge at all. The charging is something that we introduce artificially. Okay? This is the SDS. This molecule that we introduced in order to denature the protein and to add charge to it in a uniform way. Okay? So, in a sense, what you need to remember about gels, and especially, so you can, we learned just two classes or two types of gels in general. Okay, we talked about gels that separate according to size, okay, which is also true in respect to this SDS and also DNA, but the difference is that in DNA we didn't talk so much about it, but just for the sake of it, DNA molecules are negatively charged naturally. So we don't need to add any SDS. We can just take the DNA, so it has an homogeneous negative charge along it. Okay? So here, because you add something that is charged, you cannot separate by charge. Okay? And also it completely cancels the innate charge that this protein maybe had in some of the residues or something like that. And we also learned about separation by pH and isoelectric point and stuff like that.
So if it's all charged uniformly, what changes how it would go through the gel?
So the actual gel, what happens is that large molecules will have trouble moving along the gel, and thus they will move slower. Okay. We talked about liquid chromatography, about separation. So there is separation in gel electrophoresis. This is a type of separation that you can do, but there is also a different type of separation which is called liquid chromatography, which is based on affinity, of binding of molecules to specific components. Where was it? This was in protein structure and function. You should know what there is in the slide. Okay, there are 3 slides here. Okay, you should know them. You should understand what happens in chromatography. Okay, in this method that is called chromatography, which is dependent on in this case, the gel filtration chromatography is just dependent on size. In this case, it works opposite of the gel. It's like the large molecules come first because the small molecules associate to the beads that are inside this column. And in this case, it depends on Ionic interaction. Again, that this is a positively charged gel bead, so the sort of negative ions are attached to it, and just the positive ions are passing. And this is based on antibody, for example. So it has like affinity to a specific type of protein that it knows how to identify. Okay, so you need to go over the basic methods. I will not ask you what type of interaction there is between the antibody and the protein that is loaded to it, like for example. Okay? Which, yeah. You need to know what is written like here. And also gel. We talked about a little bit about gradient and centrifugation.
We should know what models, I mean, we show in the slide that models sometimes should we know the
Again.
should we know that if the mitochondria is not dense, that
Mm-hmm.
Just that it's different, you just know the ratio.
I mean, you said you should know everything it looks like, but
Again, everything is in. You said that there is a mitochondria. Where here? About separation, yeah, cell compartments. Okay, so let's see, and I think I will understand more. This, right? Cell architecture. This? No. Like, to know what type of sucrose density gradient you need to use, this is like technicality, okay? What? To know, like, we didn't talk in any stage about the actual concentration of the sucrose that we're using, okay? And also, in respect to sucrose density gradient, the only thing that you can go over, what's very helpful is going over the recordings of the class, and then you see like how in depth I get like about these things in general, and specifically about sucrose, we just presented it. Okay, we just said that this method exists and it can separate according to density. Okay, so it besides like. It doesn't separate according to like weight or size. It's like it's a combination. It's specific. It separates according to density. That's what you need to know. And that's what you need to know about sucrose gradients. And also, you can. It's good to know that like larger elements precipitate at lower speeds and lower forces. Okay, but this is in an intuition. It's also easy to understand that if you have something that is heavy or large, then it precipitates faster than something that is light. The chemical equilibrium part, what we talked about? Sorry. Okay. We can take a break if you want, but yeah. Yes. What is bothering you about chemical equilibrium in general? When we talked about dissociation concept, it's just the rate in which it's opposite of the equilibrium constant. Like the equilibrium normally describes like one direction, and the dissociation is just it describes how much will two products tend to dissociate from one another. Meaning that, so in this sense, you just have to know what it means. If you have a large dissociation constant, then the products it will tend to be in this state, right? If you have this element and this one, this P and this D, you have these two components. So if this is large, then it means that there's the equilibrium will be in a state where you have more of the products than the I don't want to use products and reactants because again it's confusing because you can switch between them, but if you have like a component A and component B that if you have here A and here B, and in equilibrium you have A, B, let's say they're connected, okay? So this is like let's say a bond, okay? So let's say there's a steady state or there's a bond between these two. So if we're talking about the dissociation constant and I tell you that Kd is larger than 1, okay, then it means that this arrow is stronger than like that in steady state. Actually it's not true, I don't want to say that, but in steady state you will have more of these elements than of these elements. They will tend to dissociate more than they tend to associate. Okay, it describes it normally describes the affinity of one molecule to another, and when it's small, then there's large affinity, and when it's large, there's less affinity. Okay, so what's important to understand in chemical equilibrium is, like we discussed also, that there's always a state, there's always a steady state, and chemical equilibrium is a specific, like if we know the concentration that of the materials that we put inside our solution, and we know the chemical equilibrium, we know exactly after an infinite time, but we will know exactly how much we will have of components A or components B after a specific time, because this ratio is the equilibrium. Okay, that is dependent on the initial concentration, but also, so also on that.
Just one thing, could you please upload all the PowerPoint presentations in PDF?
Okay.
Sometimes there's problems with, I don't know, formatting, open office,
Okay.
viewer, like PowerPoint viewer.
Okay.
Like pictures get lost or.
Okay. Download it. Yeah, it's okay. I can. So you're saying PDF, and you want questions about the methods? Okay. I'll also give you another hint is that if everything that I said that would be in the material don't assume that if we learn something in the end, then it will not be in the material. Apart from the fact that I said exactly until what.
Like after the factor.
Yeah, what I said won't be in the material will not be in the material. But what I didn't say will not be in the material, don't assume that it won't be in the material. Okay? If you assume that.
I
Yeah.
think that's a question that might sound pessimistic, but just makes me calm to know. Is there when is that? Is there no?
I don't think there is, actually.
Well, there has to be one by law.
No, I don't think there is. I think in advanced degrees there are no modes, but that's the.
I got an email about more from the risk guidance fund.
I think in second degrees there are no mode bit. No, they changed it. There's no mode bit. No. Yeah.
Or we do our exam after activity.
And what would that have? With your other exams?
No, but it's just a general knowledge.
No, look, I know that there's a lot of material, okay, for this. No, there's a lot of material for this exam. We try not to be. Let's say we will ask about the large things, okay, but so there will, if there is a specific detail, it means that we talked a lot about this detail in class, okay, and also it repeated in the slides. So don't worry, yeah.
About the cases, but there are a lot of ones that kind of,
Yeah.
So this is the same KD as the KD.
Yeah, it just you can measure the KD. We didn't discuss that, but in the book they discussed how you measure like the KD, and KD is like the point in the rate of the reaction where you have half of maximal like in the state where you have the enzyme that is maximum active. Then this is the method to measure the actual KD of each enzyme.
Here's the concentration ratio, and there it's just the concentration. How am I?
No, it's always the KD is always the same thing, but over there it's just a method to estimate it from experimental data. Okay, so it's always like how is it called, the tendency. Yeah, that's what I'm looking for. Is the tendency of when we look inside a test tube, when we look inside a solution, for the components to be in a dissociation state or to be in an association state. This is what it describes. Okay.
There's also like maybe case alpha.
There are a lot of different like constants, but this is another thing. I will not ask you something that was only in the book and was not in the presentation. Okay. So we didn't teach a lot of things that are in the book. Part of them are not updated also. So like the first chapters, they're very good in the book. Okay, so everything there is correct, and you can read about hydrogen bonds. I think they explain it probably like in Ionic Interactions better than I can explain it. But I think I showed you. Did I show you? I'm not going to go back to that. Okay, so because we talked about that a lot. Yeah. Yeah. Yes. For our case, it's the same thing. It depends. A plasmid is like people use that in different names in respect to their purposes, to what they do with it. Okay. Vector a lot of times is an element that you use to introduce something to something, or you use a vector to, like, you can imagine like a vector is something, directionality is that you introduce something. In a plasmid, a lot of time, the name plasmid comes from the actual bacteria. Okay, so the bacteria has plasmid of its own, which are circular DNA. Okay, but people misuse or do a notation abuse, like we're going to say, of these two components a lot of time. Okay, mostly people, I think that vectors are not very often used today. Like a lot of time, people are just saying plasmids.
Just to make sure, like we did talk a little about DNA sequencing and then about like evolution, but like the last thing for the test is the recombinant DNA, like the cloning.
The cloning, yeah. Yeah, I'm not gonna, although it's really, really interesting, okay, and it's like a revolution in the field, but I guess that you will learn a little bit about, I would like to talk about DNA sequencing and RNA sequencing because it's a very interesting and important technology. I hope that after this class you will be able to like read it and maybe understand it like for yourself, the actual what actually happens and what is the objective of these methods. Okay, so so there is cellular sequencing, which is like the basic type of sequencing, which started this whole thing. But today, with what we call next generation sequencing, normally what people mean is that you can get information about all the molecules, all the specific molecules in the cell, or in the sample. So in RNA sequencing, for example, you can know the transcriptome. You'll hear like when people say they mean everything in general. So it's like transcriptome, you get the whole transcript, like all the expression of all the genes inside a specific sample. The genome is the sequence of all the genes, or When people say synaptosome, for example, or a transcriptome, so it's like all the genes or something in the synapse. So everything is like that. Okay. Yeah, and again, so notice not to spend too much time on things that we talked a little about. And so for example, when there can be a question about sugars, like there was in the exam, but we didn't talk so much about it, or on polysaccharides. So there could be like a specific there could be a specific question about a specific slide, but again, we will try very hard that everything that all the questions that we give you are things that are central to what we learned, and I think that are important for you to know in your academic career, like in that sense, okay? Because so you can understand that the valence electrons, again, when I talked about like that, it's important to understand the difference between the bonds, but it's not important for the sake of this course to understand exactly what happens and what differentiates exactly between a like hydrogen bond and a covalent bond, or you need to know like the basic differences between them, and especially where these are or where they're relevant in the sense of the biological molecules. Okay? Another question? So do you think you need after you go over the materials, you will probably have more questions, right? Could be. So what? There's a forum, right? But I see that you're not using it. I think nobody asked me any questions through the forum. But if you want, then we can. I don't mind like scheduling a time on Sunday or something like that. So is there a specific time that you want? Are you free the whole time? What?
Not too early.
Are you free on Sunday in general? Like, when are you available? It's 10 to 12 and then 1 to 4. No, I don't want you to skip class, but we'll keep it short. Okay, so let's do something like this. Okay, which normally is accepted, is that in the rehearsal class I will just answer questions that you gave me in advance. Okay, and this is how we will focus it to a specific. I don't think we'll need more than an hour. Okay, so when you go over the material and you have, first of all, you're free. You can ask in the forum anytime, and everybody will see it and we'll post it. So it's great. But if you have a specific question that we want to discuss about on Sunday, so let's say on Sunday on 12, it's okay? Okay, so on Sunday on 12, and if you have, but initially for the first hour, and if you want to, we can stay a little bit longer, but not more than that, we will just discuss questions. I'll look where I can do it. Maybe we can find another class, but I will look at it now. Okay. So yeah, we'll find. We're not a lot of people. I think we'll find something. Worst case scenario, we'll do it in my room in the lab. Yeah. So we can all fit, or we do it in the grass outside or something. So let's say Sunday at 12, and then I'm just answering questions that I will be asked in advance. Okay? Okay? שמה, מה זה הבסיסים כאילו? מה זה אומר?
לא, מה הבסיסים? אני מניחה שאנחנו אומרים את זה. השאלה היא, כאילו, שאנחנו מגדירים אותם דגל רצפים של אנחנו לא חושבים על הרמה.
אני אפילו לא יודע אם יש משהו במצב כזה. יש מצב שאנחנו מזכירים רצף ספציפי של נוקלאוטידים?
כן,
שיש כאילו, שיש באינטרון נגיד, שיש A, G ודברים כאלה? לא.
כי יש לי עוד שאלות, אבל...
את לא רוצה להכיר את זה?
לא, ברור, גם אני צריכה הפסקה כאילו.
אוקיי.
אז...
כן?
אז הפסקה כאילו אולי שנייה, ואז אני אשאל אותך...
כן? מתי?
רגע, מתי מה?
לא, כאילו עכשיו. אפשר, עוד 20 דקות.
כאילו שאלה מאוד מפגרת, סתם כי השיעור הראשון כאילו לא הייתי בו, ואז כזה נשאר לי ממש בערפל, כי אני
כן,
לא זוכרת
כנראה משם הגיע השאלות.
כן,
כאילו פשוט עברנו על... כן.
אז נגיד הטבלה שכאילו בדיוק אמרת כן להתרכז בה, אז נגיד כתוב שם את מספר הקשרים הקובלנטיים שכל אחד מהאטומים יכול ליצור, והם לא כולם אינטואיטיביים, כאילו פוספט נגיד שיכול ליצור 5.
אז נתתי לכם גם רמז שבעיקרון אנחנו מדברים על, שאנחנו דיברנו על זה, אז דיברנו בעיקר על... דיברנו בעיקר על, זאת אומרת, על הדברים שבערך הזכרנו בשיעור, זה הזכרנו הרבה פעמים חמצן, מימן ופחמן. כאילו, אני חושב שכל ביולוג, כאילו כל מי שמתעסק כאילו בביולוגיה באופן כללי, אז חשוב לדעת כאילו איזה כאילו backbone יכולים להיווצר כתוצאה מחיבורים של המולקולות.
כן, הגיוני, כאילו לחמצן כזה, חסרים 2 אז כבר צריך...
כי אני מסתכלת על זה מבחינת זה, אז יש לו את השניים האלה שהם בעצם יוצרים את האינטראקציות עם המימן שיוצרים את המים, ואם את זוכרת שיש שני אינטראקציות של... ואת זוכרת שחמצן יכול ליצור 2 קשרי מימן, למשל, אז את יודעת שזה כאילו לפי הזוג האלקטרונים.
סבבה, אז זה כזה. אוקיי, טוב.
ספציפית משתמשים באותו detection, זאת אומרת משווים בין, לוקחים בג'ל, זאת אומרת זה בעיקר פיוריפיקציה, זאת אומרת אתה לא יכול לזהות שום דבר מתוך זה, כל השיטות האלה של הכרומטוגרפי, זה נגיד, זה הכל, זה כדי לזקק משהו ממשהו אחר, וצנטריפוגה גם כן. אימונובלוט זה כבר בשלב של זה בשלב של הדטקשן, אוקיי? כי על בסיס זה התוצאה שלך בסופו של דבר, זאת אומרת, אתה רוצה לדעת כמה יש לך נגיד מאותו חלבון, אז אתה משווה ואתה תראה שבמקום אחד יש לך, נגיד אם זה הג'ל, יש לי פה ג'ל, אז נגיד בנוקאוט, כאילו בעכבר מסוים יהיה ככה ובעכבר מסוים יהיה ככה, זאת אומרת, לפי העוצמה שלהם אתה יכול לדעת מה הכמות של החלבון שיש לך. זה בווסטרן בלוט, זאת אומרת זה בעצם שאתה לוקח ואתה רוצה לדעת כמה חלבון יש לך.
אבל זה עם האנטיבודי.
נכון.
אבל בקודם שהראית, איך קראו משהו בגלל זה,
זה.
זה כן.
נכון.
אז זה דטקטים על כלום.
אז משתמשים בג'ל הזה, ב3-dimensional gel בשביל לדעת את החלוקה של כל החלבונים, איך הם מסודרים, איך הם מסודרים כאילו מבחינת התא. בגלל שבנקודות האלה אתה יכול לזהות אלפים, אתה בעצם לא מזהה את החלבונים ספציפיים. אתה עושה את זה כנגד. אז זה לא שאתה פיוריפיינג, זה רק מצב שאתה רוצה נגיד להפריד את זה, ואז להשתמש בזה אחרי זה למשהו אחר. פה, כשאת רואה נגיד תמונה, אז בסופו של דבר מקבל תוצאה, מקבל כאילו איזושהי אינפורמציה, זה כבר דטקטים. האמת היא שזה יותר מתאים לקרקטורייזינג באיזשהו מקום, אבל זה כאילו דטקטים של, נגיד אם תסתכלי על תמונה כזאת, תמונה של נגיד עכבר ווייטה ועכבר ניסוי, אז יכול להיות שהגצים את שניהם אחד על השני, אז את תראי בעצם הבדלים ביניהם, ואז את יכולה לדעת, נגיד, שיש לך, שחלבונים מסוימים השתנו, או שיש לך יותר מחלבון מסוים, או שיש לך, פשוט בגלל שזה דו-ממדי.
אבל אני לא יודעת איך חלבון, אני יודעת שהם משהו שהוא יותר שלילי וגדול.
נכון. ואז אחרי זה, מה שעושים, את יכולה כאילו לקחת את האלמנט, מה קורה? את יכולה נגיד לקחת את האלמנט הזה ששונה, לבודד אותו מתוך הג'ל הזה, ואז תשים אותו במאס ספקטרומטרי, שהוא יכול לעשות אנליזה של מה בדיוק הגודל, נגיד יש לו, זה כבר קשור למה שדיברנו עליו נגיד פה, שנגיד מאס ספקטרומטרי זה אתה יכול לקבל ממש את הפרופיל של איך החלבון הזה נראה. זאת אומרת, אתה יודע לפי הפרופיל שזה החלבון הזה, לפי הפרופיל הזה זה החלבון הזה.
מה זה הפרופיל? כאילו, עברתי לך על זה, ואני לא זוכרת
את
מה הקריסטל גרף?
לא, זה לא קריסטל גרף, זה מאס ספקטרומטרי, זה מלטיטופ, מה שנקרא זה. את עוברת גם על הסליידים כאילו בצורה של זה.
אוקיי. אבל אני לא יודעת, זה לא... למה מודע? או דילגתי את זה בטעות, שזה לא היה לי.
בואי נראה, אולי זה לא היה.
היה קריסטל גרפים והיה איזה MRI.
היה מנהר? אוי, זה לא המצגת הנכונה.
כן, אני זוכרת שאני לא יודעת.
טוב, בסדר, אני אבנה את זה, אז בוא נגיד שזה לא יהיה. כאילו, דברים, אם יש משהו בשלב הזה, כאילו במודל שהמצגת ישנה יותר, אז אני כבר אשנה את זה. פרוטין. דיברנו על מה.
כן.
אוקיי, זה לא המצגת המעודכנת, אבל לא משנה. בסדר. אז כן, אז מה ספק זה...
קריסטלוגרפיה זה מה שיעשו
ב-characterizing. אפיון, כן. כן, קצת חבל, אבל בסדר. מה?
אפשר להתחיל עם תהיות.
כן.
אפשר לדעת ארוחה שלה.
אוקיי.
הדבר הראשון, אני רוצה לשאול, זה בטח שחייב להיות ניוטון, או שזה יכול להיות כאילו...
לא, בטח שהיא לא חייבת. זאת אומרת, בטח שהיא יכולה להיות גם בין נפרדים כאילו שהם...
כן. אם יש לי בתוך, נכון אם יש לי בתוך, כאילו אם זה פרלל, וזה אותו אחד, אז זה אומר שיש איזה משהו מטורף כזה ביניהם, כאילו...
שצריך להיות איזה סיבוב שם ביניהם, כן.
כן.
כן, כי הדרך היחידה שזה...
פרלל במיוחד, כאילו...
פרלל.
פרלל.
כן, באנטי פרלל זה פשוט יכול להיות ככה.
כן.
זאת אומרת, זה אנטי פרלל, ופרלל, אז זה צריך להיות איזשהו, כן, איזשהו, כאילו זה במישור של, זה צריך להיות כאילו, מין לוף כזה שהולך ככה,
כן.
ואז עוד פעם.
אבל בגלל שלא חייב להיות את הקשר הזה, אז אני לא יודעת מזה שפרלל ואנטי פרלל כלום על ה...
לא, לא. אז אתה רק יודעת כאילו את האוריינטציה ואת הסוג של ה... כן.
ואני קצת הבנתי, היה את הליביקוויטים הזה?
כן.
היה לי סקר, כי אני חושבת שבסרט היה משהו שונה מהשאר בסקר, כאילו היה
שמה?
בשלב שלא היה משהו כזה. רגע, זה הזה שעושה דגרדציה לחלבונים, נכון?
כן. נגיד, מה שאני מצפה שתדעו בהקשר של יוביקוויטין זה את המנגנון הכללי ומה זה עושה. זה יותר חשוב, כאילו, מה זה יוביקוויטינציה בכלל. אז למשל, בשאלה פה במבחן,
זה לא עושה מבחן.
אז נגיד, during the ubiquitination process, as proteins are phosphorylated several times, כאילו, אם אתה זוכר שיש שם proteins are marked with degradation target several times, what's up? No, she's just asking me questions like specific.
Yeah, substances.
Yeah.
And they said they were email for several times.
Okay. We can meet us if you want.
Really?
Yeah.
This can be really good.
Okay.
But I need maybe more days
To go over the yeah. Okay, great.
Thank you very much.
Okay.
Degradation target כאילו why is big in the degradation target?
The protein that transported to the proteasome.
כן, זה נכון.
Protein is transported outside the cell. אז מה יתנה פה?
רגע. לא, זה לא חסר. רגע. אומייגה. לא, יש את הליבוזומים, זה פשוט. הם מחוץ לזה. נכון? הגנה... ליבוסים? ווב?
נכון.
אני לא אצטרך לדעת חסר.
כן, כי בגלל מה שאנחנו דיברנו עליו, ה-ubiquitination process, אוקיי? שימי לב שאני לא מדבר על protein degradation, תקדימה שאלה.
כן, נכון.
באופן כללי. אז כי בפרוטין דגרדיישן, אז באמת את אומרת, כי השטח של הלומן של הליזוזום, זה בעצם כאילו, הרבה פעמים התייחסנו אליו בתור חלק שהוא כאילו מחוץ לתא, הוא בעצם בתוך התא. בפועל הליזוזום מפרק את זה בתוך התא, ויש גם ליזוזומים שמפרקים מחוץ לתא. אבל זה שאלה יוביקוויטינשן. זה כאילו, ליזוזום ויוביקוויטינשן זה שני פאסווייז שונים לגמרי של דגרדציה של חלבונים.
רגע, אבל אני אמרת שמחוץ לתא, אם אנחנו מדברים רק על חלבונים, מחוץ לתא זה ידי ליזוזומים, ובתוך התא זה ימיים.
כן, הרבה פעמים חלבונים שנמצאים מחוץ לתא זה ליזוזומים שמפרקים אותם, וחלבונים שנמצאים כאילו בתוך התא ולא מוגנים איזשהו מברנה זה יוביקוויטינשן, בגדול. אבל יש גם מצב של פירוק כאילו של דברים מסוימים בליזוזומים, נגיד מיטוכונדריה, היא כולה מיטוכונדריה ישנה נגיד מתפרקת בתוך ליזוזום. גם ראינו את ה...
זה לא חייב להיות עצמם או משהו? שאולי אני מבזבז?
זה משהו, לא דיברנו על זה כל כך, אבל יש את ה... שדיברנו על ה... איפה אני חושב? דיברנו על זה פה, אני חושב. כן, אבל כאילו היא יכולה לעבור כאילו secondary lysosome, כאילו מפרק את הזה, אז כאילו בהקשר הזה לא ממש ספציפית, זה חייב להיות כאילו שכל החלבונים עוברים דגרדציה, נגיד פה יש חלבונים שנמצאים בתוך התא, אז הם כאילו עוברים דגרדציה, כאילו בסופו של דבר הכל עובר כזה ליזוזומים בריסייקלינג של אורגניזם, אבל שוב, אנחנו שואלים דברים ספציפיים. השאלה הזאת על ubiquitination, אז חשוב להגיד שתביני את המטרה היא שתדעי נגיד שמה שקורה שם זה לא פוספורילציה, כי מה זה בא כאילו לבדוק? זאת אומרת, אם תחשבי שזה פוספורילציה, פוספורילציה נגיד זה משהו ממש, למדנו על כל מיני דברים שקוראים פוספורילציה, למשל בקורס, אבל זה ממש לא התהליך שקורה כאילו בדגרדציה של חלבונים. אני חושב שפוספורילציה תורמת לדגרדציה של חלבונים, אז זה כאילו זה לא קשור בהקשר של ubiquitination, זאת אומרת שאתה לא מבין מה זה התהליך של ubiquitination. מה זה פוספורילציה?